Hi friends, EnteMicrobialWorld welcomes you. In this video, we are going to see what is Gnotobiology, Gnotobiotic Germ Free Animals, how are Gnotobiotic animals produced, uses or importance of Gnotobiotic animals, terminology related to Gnotobiology, difference between Gnotobiotic and germ-free animals. Difference between germ-free and specific pathogen-free animals. What is Gnotobiology? Gnotobiology is the study of germ-free organisms in order to explore the impact of microorganisms on physiology and diseases. Next, we are going to see what are gnotobiotic animals or gnotobiotes. Animals that are either germ-free or that live in association with one or more known organisms are said to be gnotobiotic animals or gnotobiotes. The word gnotobiotic is derived from the Greek words gnotos and biota, meaning known flora or fauna. So, a gnotobiote is an animal with a known flora or fauna. The term gnotobiotes is also applicable when a microbial flora does not exist or is not detectable. Let's see what is germ-free animals. Germ-free organisms are organisms that are devoid of any detectable microbial life, including bacteria, viruses, protozoa, and parasites. Next, we are going to see how are gnotobiotic animals produced. The first germ-free organisms were chickens, obtained by sterilizing the shells of 20-day-old embryonated eggs with an effective germicide and placing them in sterile containers such as glass churn jars or steel tank-like cages into which filtered air is passed and from which waste gases are removed. Sterile food and water are placed in the cages prior to introducing the ready-to-hatch chicks. Periodic bacteriological examination of the exhaust air, feathers, excreta, and body orifices are made to confirm the absence of microorganisms in the cages or on the birds. Up to this, we learned how germ-free chicks are produced. Next, we are going to see how germ-free mammals are produced. Germ-free rats, guinea pigs, and other mammals can be obtained by caesarean section of gravid mothers under sterile operative conditions in a special chamber that allows the young animals to be introduced directly into a rearing cage. These babies must be hand-fed hourly for 2 or 3 weeks. Once established, a colony of germ-free animals can be maintained by natural reproduction under germ-free conditions. Next, we are going to see germ-free animal versus normal animals. Compared with normal animals, germ-free animals exhibit an underdeveloped immune system, making them unusually susceptible to infection if subsequently exposed to pathogenic bacteria. The normal flora microorganisms have been shown to be capable of producing metabolic products or other agents that can inhibit other microbes. For example, in the large bowel, certain anaerobic bacteria produce various organic acids like acetic, lactic, or butyric acid as metabolic waste products, which can inhibit the growth of other bacteria. Some strains of skin staphylococci have been shown to produce antibiotics that inhibit a wide variety of other bacteria. Many strains of Escherichia coli in the intestine produce colicins, which may help to protect the intestinal tract from closely related pathogenic bacteria. The germ-free animals require higher levels of B vitamins than do normal animals. Germ-free animals also require vitamin K, which normal animals do not require. These findings indicate that the normal flora may make significant contribution to the vitamin requirements of the host. Uses or the importance of germ-free animals. The germ-free animals is reared in the presence of one or more known microbial species to determine the effect of those species on growth and development of the animal or on various physiological processes. Similarly, one can inoculate a germ-free animal with a known species of microorganisms or a mixture of known species to determine their ability to produce disease or cause pathological or immunological changes in the animal. So we can say gnotobiotic techniques have been used to assess the effect of particular species of microorganisms on a host. For example, gnotobiotic techniques have helped to elucidate the role of bacteria in causation of dental caries. Thus, the gnotobiotic animal is potentially a valuable tool for investigating any suspected interaction between the host and its associated microflora or between different components of that flora. Germ-free animals are used in research involving various fields like toxicology, pollution control, autoimmune disorders, drug metabolism, genetic expression, vaccine tests, etc. Immunology is another obvious field in which gnotobiotes can prove valuable. In the study of nutrition and metabolism, Gnotobiot has made a significant contribution. Next, we are going to see terminology related to gnotobiology. Germ-free organisms. Germ-free organisms are organisms that have no microorganisms living in or on them. That is, germ-free animal is entirely devoid of any detectable microbial life. This includes viruses, protozoa, fungi, parasites and bacteria. Axenic animals. Axenic animals are gnotobiotes known to be free of all detectable microorganisms. That is, axenic animals are completely free from other microorganisms. Here, we should note one important point, that is, germ-free is the historical term utilized over the longest period of time. Its definition is the same as axenic, specific pathogen-free animals. In specific pathogen-free animals, a moderately normal microbiome is present, but devoid of specific pathogenic microorganisms. That is, specific pathogen-free animals are those from which a defined set of microorganisms, usually pathogenic bacteria, fungi, parasites, and exogenous viruses are excluded. Defined flora animals. Defined flora animals are maintained in isolated environment and are intentionally associated with one or more known microorganisms. Conventional animals. Animals reared in a room with an unknown microflora and unknown disease status. Next, we are going to see the difference between gnotobiotic and germ-free animals. When non-microbiota are introduced to a germ-free organism, it usually is referred to as a gnotobiotic organism. For example, when the intestine of a germ-free animal is deliberately colonized by a single strain or specific consortia of bacteria. Germ-free organisms are organisms that have no microorganisms living in or on them. Gnotobiotic animals differ from the germ-free animal in that there is detectable microbial life, but this is fully characterized. A germ-free animal is entirely devoid of any detectable microbial life. This includes viruses, protozoa, fungi, parasites, and bacteria. Finally, we are going to see the difference between germ-free animals and specific pathogen-free animals. Germ-free animals are animals that have no microorganisms living in or on them. Specific pathogen-free animals are free from certain non-pathogenic bacteria, parasites or viruses, but otherwise as a normal microbiome. I am concluding this video here and see you soon in my next video. Thank you, friends.